Senior tumewahi kwa bado hawajaja. Hi. Are you the technical person? Sorry? Are you the technical person?
Someone who will chair the presentation.
You are the chairperson?
No, I'm not the chair. I'm not the technical support.
You're a presenter.
No, I'm not the presenter. I'm
that's
just
difficult.
participant. I'm the technical person.
I wanted to confirm our presentation is there.
Yeah.
What is your name?
James Chacha.
That's great. Then I can change
Yeah.
mine.
And we cannot have the notes. Cannot be during projection. Someone cannot see the notes. You understand?
Like, for example, like you want to write something?
No, like I want to see the slides.
Could you check if mine is there, please?
Okay, yeah, check first, please. If the slides are
I have only 3, but we have hyperlinks. I don't know.
Let me give you mine.
Is this?
Yeah, this is mine. Yeah.
So do you want to stay with this
No,
or
no.
you want to change?
Check this ones.
And you?
You want?
What is your name?
Yeah.
So I think I don't have yours.
Oh, I uploaded yesterday.
Sorry, yes, it's written. So you want to go with this one?
Yeah,
Not this one.
not this one.
Ile.
Then you can check the.
Kama ile.
Then you come in that one.
Adveni kama hizi nzuri sana.
BMLC one,
Eh.
okay.
Iko hivi. Ila je,
Uliingilia sehemu kama ile. Uliingilia nyuma nikaona kama vile. Mbele kuangalia vizuri.
hizi ndo venye zenye rafu.
Umeona?
Hebu check kwanza.
Kule kwingine kule. Hata mpanda ukidondoka unaonekana. Huku ukianza kuchomoka unachomoka mwenyewe tutaweka miguu kwenye barabara.
Unajua ni kama
So you are having
Kushoto sio
mwangu uko katikati ya.
umetulia kama una moyo.
Presenter view.
Nimewacha roho.
Ona nilivyojichora sasa. Yaani hata sijajua.
Do you know German?
Kuna ile peni pale kumbe. Lakini haionekani. Kitabu ile naona. Yaani sina macho.
Sijui atakuwa anatafuta nini. Hawa alokata hapa wale.
Yaani nimeshtukia tu hapo ile peni nyeusi fulani.
Well, check his slides first and then we'll try this. Okay. Kwa sasa flash kwanza alafu tajaribu hiyo kitu kama kinashisha maisha sana tena ukiwa kwa mbele. Punguza stress.
What is your name?
Ewad.
So you have the 4th one. You have the 4th one.
Yes. PDF for started 3B. It will be. It is in PDF. Today, yes. There's no problem in PDF, right?
No.
Oh, it will be PowerPoint.
Yeah.
So maybe we try that thing.
Okay. So
Nimepata hapa na flash yangu.
So yeah, I want to
Natafuta flash.
Okay.
Nitafuta sana kwenye
Serious?
mama.
Using the small screen. Maybe we try.
right here.
Naona notes zako chacha.
Ndio alikuwa anajaribu.
Kama zinatokea.
Natakiwa akiweka kwenye ile ya kuweka.
Ya kuweka
Speaker view. Which one is this?
Yaani unaona huku wanaona tu PowerPoint,
Yeah.
unaona inayofuata next slide.
hivi ikiwa PDF inaonyesha kweli? No, mimi sio.
Wewe hujaweka?
Yanasemea kwamba ikiwa hiyo ni PDF au ni nani ile?
So does the online person see this?
Wanaona.
Do you know how to get to it? Kama unajua kuitoa. Have it.
Kuna namna hata mimi mwenyewe nimesahau.
The guy will come and maybe you'll assist him.
So this is in presentation monitor?
Yeah.
Do you have a pointer?
No,
Yeah,
every project presentation monitor.
Was it this
Yeah,
one?
this one.
So how do I keep this away from online people?
Not only online people, even here, we see.
Yeah, there's a way of getting rid. But if it doesn't get, I'll go just normal. I'll just go
Because
normally.
we are sharing the full screen, and if you like whatever sees here, whatever it is here. It will see on the screen.
Okay, no problem. If it this way as well, like just keep going.
Again to see.
Hmm?
Okay.
The speaker, this one?
Yeah.
Yeah. You're also using this?
I think so.
There's a guy who can assist, I think, until it.
He's not there yet.
Yeah, he's not here yet.
Okay.
So maybe he helps. But if it doesn't happen, I'll just go normally. But this way, my life is easier. Let him come first and we'll see. You're number one, yeah?
No, the last one.
So the first one is not here yet.
The first one is online.
I think so, yeah.
Yeah. Kwanza. Kwani ingie chooni hapa ni
Yeah, but sorry, I've
Na mimi ngoja niende. Hizi rangi zangu mbona ni-
Kitaka kuchora eh? Went after venue. Unataka mbele?
Nataka hapo
Katikati.
yaani hapo mstari wa pili.
yeah.
I think you are the last one.
Yes.
Is it okay now?
Yes. Yes, I think.
Thank you.
Thank you very much. So it's hybrid, huh?
Yes,
Okay.
there will be, the first presentation will be online,
Okay.
but he has not joined yet. I don't know why.
Looks like the words are not clear, or it's my eyes.
Yes, I think.
I don't know, maybe it's because.
Oh, okay, maybe.
Yeah.
So through the application, can I get the link?
To join?
Yeah.
Yes, I think, yeah, you should get it from the application.
Okay.
Vielen Dank. Oh, wir sind ja viele. Hello. Good morning, how are you? So the presenters are there, but we don't have visitors yet.
Are you sharing this?
Yes. There has been a change in shares because something has come. And we do have 1, 2, 3, 4, 5 presenters. So, Ogalo is not here. Dave?
I think the first one is
Erasmus?
Hello. Good morning.
Is there online, right? Erasmus? Are you
I'm
also
Ogalo.
online? Erasmus is not online. We can check. Nice to meet you, Ogalo. Edward.
yes, yeah.
And Mahmoud Khan, Wilfried. Sorry about this. Maybe I should call you soft cheese. It's easier, sorry. But you may have the same problem with my name, so don't worry. So if
So I think the
Erasmus
two of them,
there,
yeah,
we
two of
can
them-
swap
two of them are online.
two.
Yeah, the first and third.
Edward. No, Erasmus, but Erasmus is not online.
It has not joined yet. Ogalo, can you hear me?
Yes, I do.
So the presentation?
Yes.
So you were, and you were, you were,
Yes, pronounce.
I can read.
Wilfried, yeah.
So Edward is not here yet.
He's
Just seen him
outside.
The first one, I don't have it.
So can you ask about just in case the network is.
Yeah, let's try the.
Ogalo, do you have your presentation with you? Because I do not have it.
Yeah, it's not working.
Ogalo, are you there?
Try like this. It doesn't like go freestyle.
Ogalo?
I can assist.
Yes.
So can you present with your laptop? Because I don't have your.
So we don't have the
Oh.
time. Okay, let's.
Can I share through an email?
Okay, I will let you.
Sorry?
She will present our community visuals.
So, you know, this is,
Then.
yes, I will try. Yeah, sure. Yeah, at the moment, it's a shared screen, and what you can do is we don't have a shared screen. What you need to do is we'll get out of the presentation. And I I'm open.
You should know your presentation.
Yeah.
You may take your laptop next to it to click both that, but you need to get online. So can you ask Ogado to share the screen?
Ogalo, can you share your screen? No?
Yeah. Able to see my screen?
Please go in presentation mode so that we can immediately start.
Presentation mode.
Yes, you're not in the presentation mode yet. We only see your working environment.
Yes.
So, yeah.
Is it there?
Yes, thank you very much.
Yeah.
Just, good morning, everyone. Just a cross check. So I saw James, I saw Wilfried, Erasmus is supposed to come online. He's not there yet. Edward, you're there. Okay, and you have uploaded your presentation. Great. So I think you have, oh, this what, which was the correct? We have 2 more minutes, so please don't rely on that one. That clock is rather late. Jumping into this oral presentation, sharing, you have I have this discussion. Okay, so I'm not that I'm chasing you. And then you have mobile microphone for the first one, we can get it already. You can take this already. No, I'm handling this. The person that already getting involved in that. So we are still waiting for a few minutes before we start.
Sorry?
We just wait for a few more minutes before we start.
Okay, thanks. Thanks. I appreciate it. Thanks.
Yeah, let us wait for 1 or 2 more minutes because it's a long way up here to the hall. And some of the signs are missing on the way, so people may be searching for it. Hoping to get a little bit more audience.
Okay. Sorry for that. Okay.
Yeah.
Very small on the screen now. Okay, good morning, everyone. I try to be as German as possible with 1 minute delay. My name is Irma Jordan. You have heard already, I'm a German nationality, but I'm usually not in time. So I try to talk to allow more people to come. I'm actually at the moment based at the Alliance Biodiversity International in SIAD in Nairobi in Kenya. And it's an honor to step in for Dr. Constance Riebeck, who unfortunately didn't make it. I was informed about this yesterday, and we were close colleagues, so we just arranged that I would take over the chairing of this session. My background is human nutrition and home economic sciences. So when it comes to food science, you will challenge me. Those who know the difference, they will be aware about where the challenges start. And I'm looking forward to hearing learning from you. And I would like to introduce the first presenter who will present online. It's Ogale. He has already put up his screen and he will talk about harvest stage and methods affect quality of amaranthus, which is one of the neglected, underutilized species. I'm looking forward to hearing from you, Ogale. You will have 10 minutes and please keep in time. I will interrupt you and mute you in case you're taking too long, so that we have a little bit time for discussion. Welcome, Ogale. Ogalo, sorry. It's on you now. Please unmute yourself.
Thank you very much. Thank you very much. I want to thank the organizers for giving me an opportunity in this platform so that I can present my work. The title of my work is Harvest Stage and Method Affect Quality of Amaranthus. And we've done this in guidance together with Dr. Muti, Dr. Jackson, and Dr. Gogo. This is the order of my presentation from introduction, objective, materials and methods, data collection, data analysis, results, conclusion, and recommendation. A vegetable amaranth, which is commonly known as mchicha in Swahili, which is majorly grown in the coastal lowland of Kenya, and is majorly grown for consumption and for income generation, but there is dwindling, despite the dwindling production of this vegetable, there is significant loss due to harvest methods, due to post-harvest handling, environmental condition, which have got adverse effects on both crop quality as well as quantity. The optimal harvest method for vegetable at various development stages is still unclear. Hence, there is need determine the optimal harvesting method and harvesting stage for a longer post-harvest quality, as well as nutritional and yield quality of the vegetable. The objective of the study was to determine the effects of the harvest stage and method on the harvest quality of Amaranthus in coastal Kenya. Materials and methods. The experiment was set up in Pwani University, which is in Kilifi County, that is Kenya. And the two trials was done between April and September in the year 2022. The experiment was laid down in a randomized complete block design in a 2x2 factorial 2x2 factorial treatment arrangement with 3 replicates. The factors studied were the harvest stage at 2 levels, that is at 6 weeks or at 8 weeks, and harvest method at 2 levels, that is pinching or uprooting. Data was collected on the fresh weight, dry weight, total carotenoid, total chlorophyll, proximate composition, which include protein, fat, carbohydrate, energy, ash, fiber, micronutrient, which is potassium, calcium, and magnesium. For the micronutrients include iron and zinc. That analysis, the data was subjected to analysis of variance using SAS. Significant means at F tests were separated using the Tukey's test. Data was analyzed at 5% level of significance. Results. This is the summary data, and the study just show that show that there was a treatment, there was effect on the treatment, while the NS showed that there was no effect on the treatment. Amaranthus harvested at 6 weeks had higher protein content, higher carbohydrate content, higher energy content, and higher ash content, as well as fiber. And this could be attributed to at 8 weeks, the plant has got a wider canopy, which provide wider surface area for photosynthesis, hence accumulating more photosynthesis. For the zinc content interaction, Amaranthus harvested at 8 weeks had higher zinc content compared to those harvested at 6 weeks. In conclusion, vegetative amaranth harvested at 8 weeks after planting had higher fresh weight at 37.1%, dry weight at 17.7%, protein at 34.3%, carbohydrate 25.8%, energy at 15.6%, ash at 22.5%, fiber at 20.2%, carotenoid at 38.8%, iron at 34.7%, zinc at 35.3% content compared to those ones harvested at 6 weeks. And harvesting at 8 weeks, irrespective of the harvest method, will offer higher nutritional value and yield of the vegetable amaranth. In recommendation, there is need to investigate the effect of the harvest stage and the method on post-harvest quality of the vegetable amaranth. Thank you.
Okay, thank you very much, Ogal. You are actually very quick. You have 5 more minutes. And before we get into the discussion, could you go back to the section where you talk about the different harvest methods you were comparing?
Different.
You have the 6 and 8 weeks as a different harvest method?
Yes, yes, yes.
Okay, can you go back to the table? You were very quick running over.
Yes.
And then it was. You
Here
have
or
5 more minutes to explain us what you did, so just to make sure that we understand you correctly.
oh here on the experiment.
Yes.
Thank you very much for the position again. The experiment was laid down in a randomized complete block design. That's a 2x2 factorial treatment that was done in a 3x3 replication. So here we are taking samples randomly, collecting samples from each of the plots, and then we tag them, and then we send them to the lab for analysis. And then the next factor, that is the harvesting that was done at 6 weeks. So we will have samples. We pick samples from each block, and we pick them randomly, and then we harvest them at 6 weeks, and then we label them. Then we send them back, send them to the lab for analysis, so that we will understand the nutritional component of various parameters that we are studying in these samples that is collected. And the harvesting method also done at 2 levels, and that is by pinching and also uprooting. And these are just general methods that the farmers in Kilifi are using in the farm while harvesting, but they lack the knowledge to know which one will give better quality or will give better post-harvest quality after harvesting. So we are just carrying out using this method so that we could identify which could best give higher nutritional quality and yield.
Okay, so Ogalo, just one more question. So the 6 weeks and 8 weeks, it is the same plant, or did you compare a plant harvested at 6 weeks and compared it with another plant which was harvested at 8 weeks? So they were more mature in the end, or did you harvest the same plant once at 6 weeks and once at 8 weeks?
No, we harvest a plant from the different plots.
So each
we don't separate from each plot, but at different plants.
Okay, and then in terms of processing for the food science lab, did you do snap freezing? Did you dry them? Did you put them fresh into the lab? And what was the transition period?
We package them, and then we send them to the lab where they were analyzed. Actually, we're looking at the fresh weight, and then we dry them, and then look at the content as the days progress.
And the period between harvesting and reaching in the lab for analysis, how long did it take?
A day, just 1 day.
1 day. Okay.
We harvest the Kilifi, then we pack them and send them to Nairobi for analysis. Yes.
Okay. No, I think that clarifies some of my questions. Now, going back to the room, online participants, I think you are at the moment the only one. Is there any more questions from the room? Yes, I come to you.
Thank you for the nice presentation. But I'm so curious to ask him what is really the hypothesis when comparing, for example, they have said matter at 2 different stages. I mean, 6 weeks and then 8 weeks, and then comparing again the biomass on a fresh matter basis, because there's different water content in those matter harvested, and I really wonder how he managed to compare this. Also, is it a common practice in the region to uproot amaranthus? Thank you.
Thank you very much for your question. If I got your question well, actually, in the coastal region, this is where most farmers grow this vegetable for income generation and also for consumption. And they tend to harvest at different stages. So they are not sure which one will give higher value in terms of nutrition, or which one is liable to a loss of losses from the market. Because actually, this is just one of the objectives you have got, I was carrying out now, pressing it from the farm up to the market. What happens? What quantity is lost until it gets to the market? So we'll think how many is lost through vibration and all that. But actually, farmers are not sure of which is the best method or the stage in which to harvest this vegetable. So they just collected this based on the practice that's happening that the farmers are using on the ground.
Thank you. There's one more question.
Yes, thank you for the presentation. I still have concerns about the slide you are showing. When you mention harvesting method at two levels, harvesting method are pitching and outputting.
Yeah, pinching and putting. Yeah.
What about the two levels? Is the same thing for harvesting at two levels? We did not specify which levels. And when you say harvest stage, we are expecting technological stages. But here it's not, you mention harvest time. 6 weeks and 8 weeks in harvest time, not harvest stage. And the two levels of what are these two levels? It's still unclear. Thank you.
Okay, thank you.
Quick sentence.
Thank you very much.
One or two questions. You may take over and well. One sentence. One sentence.
Thank you.
I'll request that I'll request, there's a lot of echo, so I don't get the question clearly, please.
Okay, I think I hope you can understand my voice. The question was regarding clarification
very
again
clear.
on the harvest methodology. You were mentioning time only, but not the plant stage, the growth stage, and the two levels you're referring to. So you would need to clarify this a little bit more. And then there's another question coming.
Hi, thank you. I just wanted to
Thank
ask,
you.
you have two levels. You have the 6 weeks and the 8 weeks. And because it was clear for me that 8 weeks is better than 6 weeks, and I would like to ask if you see any potential in 10 weeks harvesting.
Thank you very much. For 6 weeks, actually, at eight weeks now, the plant now starts to wither and lose chlorophyll, and that's why we only stopped at 8 weeks. And for pinching, we actually harvested the whole plant just above the ground. We did not take a leaf or a what, but we only just harvested the whole crop above the ground. That's what we refer to as pinching is. But now uprooting now, we take the whole plant system plus the root system.
Okay.
If I got the question rightly.
Thank you, Ogalo. I think it needs a little bit more clarification in the abstract, maybe. It will also help us to follow up better on your results. Then there is one question online, if there's no further one here.
Yeah. Oh, I can open it.
No, there was no online.
Oh, there's no.
Thank you, Ogalo. Interesting to, you just can also contact us because we also did just like in the drylands and a field trial with amaranthus and intercrop this with cow beans. But it's yes, of course, yield increases the longer you wait for the plant to grow. That's also a criteria where you may use this as a confounder in terms of their food composition in the end. So please, can you stop sharing? Erasmus, please get prepared. You are the third one in the row. And we have the next speaker, James who will talk about the effect of biodegradable modified film packaging on the quality and safety aspects of African nightshade, another underutilized crop.
Okay, good morning, everyone.
Good morning.
Yes,
morning.
I'm James Simon Chacha. I'm a PhD student from the Humboldt University of Berlin. And the topic of my presentation today is the impact of biodegradable film packaging and storage conditions on the health promoting compounds and microbial diversity of African nightshade leaves, and this is the Solanum nigrum that we worked with. So I'll be presenting this on behalf of my co-authors and supervisors. And this work is part of a huge project work that I'm studying, is funding my study by the Cocoactive project. And this is a project that is based in 5 institutions, 2 in Tanzania and 3 in Germany. And it's funded by the Federal Ministry of Food and Agriculture in Germany. So this is the outline of my presentation, and briefly, African indigenous vegetables, or AIVs, as I may refer to them, are plants or crops that are native or found localized in Africa, in the African region. And these are usually consumed alongside the common food staples such as maize meal and boiled rice or cooked rice. So people from Tanzania and Kenya and even Uganda know ugali, so the stiff porridge. So typical examples of these African indigenous vegetables include amaranth, like the one that has been presented previously, African nightshade that I'm focusing on. You also have pumpkin leaves and Abyssinian mustard. So these African indigenous vegetables are usually considered climate resilient, not only due to their higher tolerance to a wider spectrum of abiotic stresses, but also due to their higher level of adaptation to local conditions in these, let's say, the villages and where they are grown. So they have a higher resistance to pests and diseases, and they also have a rich genetic diversity. And they are, the AIVs are income generating, and they're also nutritional benefits and health benefits due to their composition contents of micronutrients, vitamins and minerals, and the secondary plant metabolites. So they are considered important in the achievement of the Sustainable Development Goals. Number 1, no poverty, 2, income generation. Number 2, zero hunger due to those micronutrients and secondary plant metabolites. And secondary plant metabolites, the health benefits for SDG number 3, good health and well-being. But they have, due to their high perishability and due to moisture loss and wilting and all these factors, inadequacies in packaging and storage technology, so their quality and safety is being adversely affected. So briefly on MAP, or modified atmosphere packaging, this is a packaging technique that is used to enhance the shelf life of products, or in this case, vegetables. So in this specific technique, the oxygen and carbon dioxide level in the package that contains the product or vegetables, in this case, is passively or actively altered to meet the product specific requirements. And this now will affects the respiration, transpiration, and ethylene product. And in many countries globally, and in Kenya and Tanzania, there has been a ban of the conventional film plastic packages due to their health and environmental concerns. Now, attention has been shifted to the green label alternatives in form of this biodegradable film packaging, and this is where my study is. So when I'll be saying biopacks, I'm referring to these biodegradable film packages. These are considered environment friendly and non-toxic to humans. So the aim of this study is to determine the effect of the biopacks on the characteristic health promoting compounds and microbiological diversity of the African nightshade leaves and these species that we're working on. And this is during a storage at 6 degrees centigrade and 20 degrees centigrade for up to 7 days. I'll get into this description later. So this is the materials and methods. So I won't go into details of this, but briefly, we grew 65 nightshade plants in the Dahlem experimental station in Humboldt. And we harvested these. They took quite long to grow and harvest, 7 days. So we ended up testing or looking at the color indices And for the secondary plant metabolites, for the bioactive compounds, we check the ascorbic acid representing the vitamins, and flavonoids and phenolic acids, carotenoids, and chlorophyll. These are the secondary plant metabolites. And for microbial loads, we checked the microbial loads, yeast molds, and total mesophilic counts, and the Enterobacteria C and Pseudomonas C. So briefly on what we did. And we used our methods that we use in the lab with slight modification. So there are references there. If you need this, I can provide. And this briefly shows this is 4 weeks, 3 weeks, 2 weeks, and 1 week before harvest. So this is quickly just an overview of the design of the experiment. We carried quality evaluations at day 0 harvest, which represent the control, and then the treatments at day 3 and day 7, each having 6 degrees centigrade and 20 degrees centigrade. So we obtained this 6 degrees centigrade in a climate chamber. We used a climate chamber in our lab there. And so we have biopacks and unpacked. This one open plates just to get a comparison. So briefly, quickly to the results. 5, okay. 3 minutes, okay. So I'll not speak on the gas composition, or I can just say briefly about this. Yeah, so we had strong changes in the oxygen and carbon dioxide concentrations until day 3 and day 7. And this is, literature shows that due to the changes in respiration right after harvest of a product. So once the vegetables are harvested, they continue respiring just like any other products until they find that they don't have the same growth conditions again. So, but there was again changes in day 7 in the oxygen and carbon dioxide concentrations. So what we obtained indicates a passive map because we did not actively change the gas composition, just closed and the atmosphere changed itself. So it was it gave a passive environment for our experiment. So, and no more details there because of time. We also had a look in the loss in fresh weight between the biopack and unpacked, and we found that the biopacks significantly reduced the losses in fresh weight. And this is due to the fact that we used biopacks that were non-perforated. I'll go into details again. And for the color indices, so the biopacks retain the leaf color at least up to day 7. And compared, this is different to the unpacked samples. This one showed, and on the ascorbic acid and secondary plant metabolites, the ascorbic acid was degraded as days go to day 7, but the total phenolic acids and total flavonoids were enhanced or increased and at 20 degrees centigrade. So lastly, on the microbial diversity, so our passive map did not lower 1 minute or out. Okay, our passive map did not have a great impact on the microbial loads because the microbial loads increased up to day 7. So it had a little effect on the microbial quality of the preserved samples. So in conclusion, the biodegradable film packaging was found to be effective to a certain extent in retaining the quality and safety aspects of African nightshade leaves after harvest. So fresh weight losses were significantly reduced, and visual qualities, this is the color indices, were retained in the biopacked compared to unpacked samples. And the total phenolic and flavonoid contents were retained, and in some cases, significantly enhanced at 20 degrees centigrade, irrespective of the packaging condition. But ascorbic acid, as I said, was degraded, irrespective of storage conditions. So I'd like to end there, and these are the references for the parts that I used, and I'd like to acknowledge the partner institutions and the funding agency for funding this study. Thanks a lot.
Thank you very much. Any immediate questions? No. Could you go back to your treatment study flow? I have a question there. You said that the control is the one which stays for 0 days.
Yeah.
And then you have two treatment groups, one biopacked, one unpacked.
Yeah.
I'm confused there. Is the unpacked not the one which is controlling the biopacked? And why do you call it treatment? What did you do for the unpacked to be different to the control?
Okay, thanks. I'm answering.
Thank you.
So it's good to have this time again to explain. So what we did here, this control or day 0 is just right after harvesting. So the fresh samples right after harvesting, this was freeze dried and okay, it's for everyone, you are hearing, okay. So these were right after harvest, the fresh samples were freeze dried and now the analysis started. But the rest of this, we have we call these treatments, and this is the control because the fresh just after harvesting. So these are treatments, but the biopacked and unpacked. So this unpacked is not a control. We do not consider it as a control, but a treatment because we included it just for comparison purposes. So this is the aim. This is why this unpacked is not a treatment. We included it for comparison purposes. So yeah. If I answered your question, and this was fresh right after harvest, freeze dried and for treatment.
I don't consider the 0 day as a control for the 6, the 7 days, the 3 days. But there are 2 more questions. Erasmus, please keep your hand up so that we don't forget you to check.
Thank you for your presentation.
Yeah.
Please, I want to know what would be the reason for the degradation in ascorbic acid in all the treatments applied.
Yeah, the degradation of ascorbic acid, we think that this was due to several factors. First, the storage duration, and second, the temperature, and also the packaging, and also the presence of residual oxygen in the biopacks. So this provides a oxidative degradation effect. That's why there was, as the days went by, there was a degradation of ascorbic acid. But we still found the content of the ascorbic acid to be, for the biopacks, to be between 15 and 31%. It is for these 2 temperatures, 6 and 20 degrees centigrade.
Okay. Still on this slide,
Yeah,
which
I know you will come.
I still do not agree that your control is the 0 day. Your unpacked should be your control. You can relook at that. Then again, from your topic, you want to let us get the impact of modified atmosphere. Can you tell us what compositions of air went into your modified atmosphere for us to appreciate your work? Thank you.
Thanks a lot. First, I'll explain a bit here. We have the only best reference on this is Elisha Gogos, the first presentation, yeah? They did a similar experiment on modified atmosphere packaging, but with different species and different temperatures. So for them, it's a matter of thinking and checking, of course, literature. They considered, they had something like this, and they considered each, as you are saying, as a control. So I'll check into this. But for the oxygen, and it's not moving now, but the oxygen and carbon dioxide concentration, the carbon dioxide concentration changed significantly. We had a 100% increase in carbon dioxide. So from the normal 0.03%, we reached 3%. If I remember the numbers very well, you can see from the figure, if you can project it, but
no, not anymore.
Okay, so this is what we had, and I could not explain so much due to time. Yeah.
The time is the same for everyone. No, the time is the same. I can share. Yes, so please, then he's inviting you for a joint lunch to explain this. So with the next speaker, Erasmus, we are trying to put your slides on the screen, but you also had a question. Can you unmute yourself and raise a question?
Yes. Good morning, everyone. It's morning here. Thanks to the last speaker for the presentation on African nightshade. I actually had 2 questions to ask. The first question was on the experimental design that you have already addressed. The other question is on color measurement in vegetables. Using the LAB system, is that correct? Is it logical to use the LAB system to define if vegetables are fresh or not? Thank you.
If I understood your question well, that we did with these color indices, they're using the LAB system to determine the freshness. If that's the case, no, we did not use LAB system to determine the freshness, but the changes in appearance of the leaves after each treatment. So, yeah. In addition to that, So this way, just to check the changes in the appearance, external appearance in terms of greenness, lightness, as these are the indicators for these changes and not for freshness.
Yeah, but then the LAB for green leaves.
Yeah.
African nightshade presents green leaves, right?
Yeah.
Using the LAB to measure the color of green leaves to me seems a bit not very right, because remember that for the LAB system, L is measuring the lightness to darkness values.
Okay, so in this case, please take your email address, exchange this, Erasmus, and you, James, will have to communicate and let the audience know with your publication.
Yeah.
Okay.
Okay, thank you,
Erasmus, for your questions. You are now the next speaker, even. So I invite you to talk about grain shape and processing type and grain origin when it comes to in relation to mycotoxins. Because of time, we have uploaded this here already. Let me know whether I shall click the next slide. And you have 10 minutes for the presentation.
Okay, thank you very much. Can you hear me well?
Yes.
Okay, good morning, everybody, and welcome to our presentation on mycotoxin concentrations in rice, which are affected by chalkiness, grain shape processing type, and the origin of the rice. This study was done in collaboration with the Institute of Agronomy and Crop Physiology at the Justin Liebig University, and also with the International Institute of Tropical Agriculture under the framework of the Plant Health Initiative, the CGI Plant Health Initiative, and the Healthy Diets for Africa project. I am here to represent all the other co-authors. Next, please. As we all know, for those that know about mycotoxins, which are secondary metabolites secreted by toxigenic fungi, they are capable of causing stunting in children, immunosuppression, and cancers, even in elderly people, and can also lead to death. Some people even consider mycotoxin a kind of hidden threat to public health, especially in areas, in subhumid areas where grains are being produced and are also being consumed in suboptimum conditions. These grains are often exposed to contamination due to the climatic conditions. Mycotoxin management strategies are often regulated by strict laws or strict regulations, and the mycotoxins can also be monitored continuously to ensure prevention And we can also apply detoxification methods in areas where we have to do with soluble mycotoxins, water-soluble mycotoxins. One of the most important strategies to control mycotoxin is public awareness and education, which is actually lacking in most areas. In the case of Africa and Asia, where we have the highest production of rice serves as a food security crop. And so it is very important that we monitor to ensure that mycotoxin does not exceed their maximum residual limit. For this reason, please can you continue? For this reason, we seek to investigate the prevalence of total aflatoxin in commercial rice from rural and urban markets in sub-Saharan Africa, and to investigate in depth the effects of intrinsic grain quality characteristics, the type of processing and the origin of the rice on aflatoxin contamination. In addition to this, we are also going to look at the co-occurrence of aflatoxin and other mycotoxins like fumonisin and zearalenone. We collected brown, white, and parboiled rice from 54 markets in 5 African countries, and we collected a total of 527 samples. These 527 samples were analyzed for grain quality and also for mycotoxin contamination. Next, please. The physicochemical properties of all the samples that we collected are segregated here as a function of the origin of the samples, as a function of the type of processing. Type of processing here shows whether the rice was white meal, brown, or parboiled, and as a function of the type of market. And generally, we saw that for the origin of samples, imported samples had high head rice ratio. That is grains of intact grains, and also the mixed variety for wheat was highest in domestic rice. Mixed variety for wheat is a parameter here that says whether your variety is pure or not pure. So the domestic samples were not pure. We also noticed that the imported samples had lower pasting viscosity properties compared to the domestic samples. And as a function of type of processing, we noticed that the parboiled rice had the highest level of head rice ratio, which was normal. The highest length to width ratio, the grains were long and slender. And we also saw that brown rice presented the highest level of impurity in terms of varietal mixture, and that white rice had the highest level of chalkiness. We also noticed that in terms of type of market, the urban samples presented the highest chalkiness compared to the rural samples that were collected in rural markets. Next, please. When we did the stepwise regression model of the quantitative variables here are the length-width ratio, mixed variety for wheat, color, chalkiness, protein content, amylose. We saw that the length-width ratio, the mixed variety for wheat, and the chalkiness were the parameters, the intrinsic parameters that significantly influence the aflatoxin concentration in brown, parboiled, and white rice. Next, please. When we looked at the qualitative parameters, such as the origin of the samples, the processing type, the type of market, and shape classification, shape and chalky classification, we saw that when grains were slender, these grains tended to present higher levels of total aflatoxin than grains that were medium or bold. Chalky samples had highest level of total aflatoxin than non-chalky grains, and parboiled rice had lower total aflatoxin compared to white rice. Brown rice fell between parboiled and white rice. Next, please. The prevalence or the concentration of mycotoxins in all the samples that were analyzed showed us that 379 of these samples, that is approximately 72% of all the samples that we collected, had detectable levels of aflatoxins that range from 3 parts per billion to 89.8 parts per billion. Fumonisin occurred in some samples, but the detectable levels were low compared to zearalenone, which was detected in 20 samples, while deoxynivalenol was not present in any of the samples that we evaluated. Fumonisin and deoxynivalenol are aflatoxin, are mycotoxins of the same properties. And so we noticed that because of their chemical structure, these two mycotoxins were not common in the 3 different types of rice that we collected. And we saw that total aflatoxin co-occurred with zearalenone in 30% of the samples. Regarding aflatoxin alone, we saw that 47.49% of the samples exceeded the European Union maximum residual limit, indicating to us that most of the rice that is consumed in our market is actually posing a threat on health. Next, please.
You have 1 minute left.
Sorry?
1 minute left.
Okay, this is a summary. This is a summary of the study and the mitigation methods, strategies that we are proposing for the better conservation of rice in sub-Saharan Africa. Next, please. The implications of our study are found here. People should choose rice of specific land-width ratio, but this should be accompanied by the strategies that we have proposed. That is better storage in hermetic bags, and also sensitization should be intensified in our communities. Thank you very much for this opportunity that Tropentac gave us to present this work. Merci.
Thank you very much. So thank you very much, Erasmus. I will close the presentation, and if you can show your face for the discussion would be great. Oh, no, I'm doing something wrong here.
I can't see the video button to put it on.
Okay, but meanwhile, we can collect some questions for you.
Yes, I'm ready, please.
Yeah, thanks, Erasmus, for your presentation. So it seems that your study is more of a correlation than causation itself. So I'm wondering whether first, were you able to keep or maintain the humidity and temperature for all of the rice samples? Because basically, that's the main factor of influencing the mycotoxin concentrations or levels. So were you able to keep the humidity and temperature for all of the rice samples?
Sorry, please, were we looking at?
Did you hear him? Erasmus, did you hear the question?
No, please.
It was keeping the temperature same and the humidity the same throughout the process of analysis of the trial. Did you manage to do that to ensure temperature and humidity keeping the same?
Yeah, we used the Neogen kit for both the extraction and the quantification of all the mycotoxins that we analyzed in this study. And each of the mycotoxin has a protocol that comes with the kit. And these protocols are based on the enzyme-linked immunosorbent assay method that is based on the interaction, the immunological interactions between the antibodies and the antigens. The antibodies come with the strips provided by the Neogen kit. So it's actually an extraction that occurs at room temperature. We are not heating, and this extraction uses hydroethanolic hydroethanolic solutions.
Thank you very much for an insightful talk. I have a quick question concerning the causes of a lower aflatoxin concentration determined in parabolic rice. Is that due to the paraboiling process, or do you think that you got better rice to start with? So the rice that undergoes paraboiling has already a selection before, and it has nothing to do with the paraboiling, but rather with selection.
Please, if I hear your question well, it has to do with the aflatoxin concentration in parboiled rice.
Yes.
Why is that?
The question is, parboiled rice may have undergone a pre-selection. Is this one of the explanations for the lower concentrations? Or is there another explanation for the lower concentrations in the parboiled?
Yeah. The different parboiled rice samples that we collected were all the parboiled rice methods. Please, can you hear me?
Can you repeat your response again? You were accidentally muted in the queue.
Sorry, I can't hear you very well. What's happening with the sound system?
Can you respond again to the PowerPoint, because we accidentally muted you on the way. So we need your response again. The audience will be interested in the PowerPoint response.
Okay, I was saying that the parboiled rice samples that we collected were randomly collected across all the markets. And so the exact method that was used to produce the parboiled rice, we cannot define the exact method, but we know that all rice is parboiled by soaking, steaming, drying, and milling. During the process of soaking, there is a high tendency that
We got your answer. You have little pre-selection of the quantity, so you may reflect on your final results and using this as a confounder. There's one more question, and then we need to proceed. So one sentence as a response.
Please, can you take us through how you determine viscosity in rice?
Hello, can I tell you why we measured viscosity?
How do you determine viscosity in rice?
Okay, we determine viscosity using the RVA method, the rapid viscoanalyzer.
Thank you. And then there's one question from Tima.
Yeah, thanks, Erasmus, for your presentation. I wanted to understand the country. You said that for the samples that were imported, they had lower quantities of aflatoxin. So I wanted to know from which countries were these samples imported from?
Yes, these imported samples were collected in the market. And for each samples that we collected, we looked at the bags in which the samples were sold, and then we collected information on the origin. This origin here referred to countries, and these samples were mostly from Southeast Asia and also from Asia in general. Here, you know, imported rice in sub-Saharan Africa largely comes from countries like Vietnam, India, Pakistan, and even in the literature, we saw that samples that were analyzed from these countries were also presenting high levels of aflatoxins.
Thank you, Erasmus. And please link each other up with the emails. You will find the contacts in the Woover app. Also for any other follow-up questions or comments. I would like now to welcome Edward. Edward is talking about food price elasticities of demand and consumer food choice. Looking into the implications for this on the food and nutrition status in Tanzania. So we are changing the discipline slightly, going from food science now to agriculture economy or food economy, depending on how you frame it. Welcome, Edward.
Okay, thank you. Good morning, everyone.
as she has just said, let's having heard from food scientists. Now let's refresh with the agricultural economics. So my name is Iwad Mushi. I'm a PhD student at the Sokoine University for Agriculture in Tanzania. I'm going to present food price elasticity of demand and consumer food choices, looking at the implications for the food and nutrition status in Tanzania. This is another study, part of the big FOC active project being implemented in rural urban selected areas of Tanzania. We used to say our health outcomes are a result of what we eat, keeping other factors constant. So we can see that globally, the world is faced by the malnutrition problems in all its forms. The prevalence of malnutrition at the global level is that obesity is at about 15%, anemia 29%, stunting 22%, wasting 6.8%. And this is a global problem. Every country is affected by malnutrition. At least one form of malnutrition affects every country of the world. While the developed countries are affected more by overnutrition and obesity, unfortunately, sub-Saharan African and many low-income countries are affected by either double burden of malnutrition or the triple burden of malnutrition. And those low and middle-income countries includes Tanzania as well. As you can see from this map, Tanzania is located here, and it is affected by all three forms of malnutrition. As you can see in terms of statistics, undernourishment of the total population is about 23.8%, while obesity of the adult population is 12%. Stunting, wasting, child obesity are also not in desirable numbers. So this calls for more measures to control or to address the problem. We cannot more emphasize the consequences of malnutrition. We all know it results to health, economic, social, and many aspects which are not good to the society. But it's known that eating diverse food could be among the solutions to address the malnutrition problem. So this is well known. But yet, without this, despite this knowledge that we have, there is low dietary diversity. That many people do not consume foods in diverse. So the question is, if we know that eating diverse foods would help to address the malnutrition problem, then why is still there low dietary diversity? So the conflicting question could be, are the diverse food not available? If they are available, are they not accessible? If they are accessible, are they not affordable? Or if they are accessible, are the quality and safety not sufficient enough to control the problem? Or it is the convenience problem that we cannot easily prepare or access the food? Or are we influenced by the promotion and advertisements of foods that makes us choose unhealthy food? So these are the conflicting questions that we need to answer. So in this study, we did not answer all these questions. We only focused on one, which is accessibility, and specifically the economic accessibility, looking at the price levels of foods. This chart here shows the Tanzania food-based dietary guideline, which is a guideline for consumption of a healthy diet. So you can see, for one to have a healthy diet in Tanzania, you need to consume all these six groups. So asking these questions takes us to the thinking of the entire food system. So if we look at where do we fit these questions in the entire food system. So we find that they are fitting in the food environment. So this is where this study focuses on, looking on how the food environment influences the consumer behavior in terms of how people make choice and what the choices will mean or imply in terms of diet of the people. So these are two main objectives of the study, looking on how consumers respond to demand of various foods when their prices change. And also the second objective looks on how consumers respond to demand on food when the price of other food groups do change. The study was implemented in Ilala, representing an urban setting, and in Mkuranga district, representing a rural setting in Tanzania. We utilize cross-sectional data collected through the semi-structured questionnaire, and we implemented the multi-stage sampling with stratification. The study is being underpinned by the theory of consumer behavior, and we adopted the almost ideal demand system to estimate the price elasticity. I won't go into details of this, but I'll be glad to hear questions about this if they happen later. So here are the key findings. We found that animal foods and their products are the most expensive foods throughout the 7 food groups which were explored. And surprisingly, fruits and vegetables were the cheapest food groups in the study. These again have implications in terms of nutritional status. The expensiveness of the animal foods and their products say something about the source of animal proteins to consumption and the resulting health outcomes. And again, despite the low price of fruits and vegetables, still some studies show that there is low consumption of the same. So this again poses the question, why is the reason behind? And we saw also a significant difference in terms of prices, food prices across urban and rural areas. But an important thing to note, we found that in rural areas, sugar and sugar products and oils and fats are the most expensive food groups among those food groups which were explored. The rest of the groups, urban areas, prices were higher than the rural areas. Again, this gives us a conflicting question of the divergence between urban and rural areas in terms of food prices. Looking at the price elasticities, I will not discuss all of the numbers, but I've chosen few. First one is we found that except for oils and fats and sugar products, own price elasticities of all food groups exhibited a negative inelastic demand, implying that as prices of food increases, the demand for the same goods went down, which is, of course, according to the demand to the law of demand. But we got an exceptional demand. On price demand on sugar and sugar products and oils and fats. In these two food groups, when their prices increases, the demand or consumption of the same also increased, which is against the law of demand. Against this also tells something or warn us about the intervention. So this suggests that price could not be an effective intervention to reduce or to discourage the consumption of sugar and sugar products and oils and fats. Another interesting result is that we found a complementary relationship or consumption between fruits and vegetables. Practically or theoretically, we could not imagine that there could be complementarity in terms of consumption of fruits and vegetables. But in practice, this is what we found, and we can relate in the local context of Tanzania when we find that everywhere you go to buy fruits and vegetables, you will also find the two are being sold together. So probably this could be the reason of why we found the complementarity in terms of consumption between these two food groups. So to conclude, I would say that food prices for oils and fats, sugar and sugar products are significantly higher in rural areas than urban areas. Consumer responsiveness of food demand to price changes is not healthy, especially on the two main groups, which are fats and oils and sugar products. Therefore, we could say that if we increase the production of fruits and vegetables, we may reduce the prices of the same and reduce the elasticity nature of the two goods and hence increase the consumption of the same. And we may say that given the result that we got, non-price interventions could not reduce I mean, could reduce the consumption of sugar.
We can read
sugar
that last
and-
sentence ourselves. Thank you very much. We are eating on your discussion.
Thank you very much.
Thank you very much. It's a challenge. It's a real challenge also in this complex topic. Any questions from your side? Oh, okay. Meanwhile, I raise my question because I'm also handicapped with my back walking. Why did you grow vegetables and fruits? They are such a diverse food group. How do you manage to put them together? You don't need to respond to that immediately. Neither. Okay, but okay, I will not that one.
I have a quick question concerning the reasons for the lower food prices of oils and fats in the city and sugar. Has that to do with their import facilities, so that they actually come from the port directly to the cities and then are distributed to the countryside?
Okay, thank you. Actually, the reasons you provided are my response. So probably the reasons for higher prices of oils and fats and sugar products in the urban areas. One is about 40% of sugar and sugar products of sugar in Tanzania is imported. Only 60%, around 60 to 70% is produced locally. And again, the distribution of sugar in the country is centralized, and it's in few regions. So specifically in the study area, the transport logistics could be the reason for the higher price of sugar. The same for oils. For oil, we literally use. Thank you.
Yes, in line with the response you just gave, please, what is the sugar consumption pattern in Tanzania? Because you started by telling us about the nutritional values for Tanzania, and you suddenly landed on sugar. And why not any other products? For instance, meat and meat products, but sugar.
Okay, thank you. First, we didn't start specifically the consumption pattern of sugar in the country, but the reason we emphasized on sugar and oils, these two are considered as not healthy food groups among the other food groups. And we considered, I gave more emphasis on fruits and vegetables, is the vice versa. The two are considered as more healthy foods in a healthy plate or in a diet. So I could have discussed all of them, but because of time, I chose two: the considered unhealthy food groups and considered healthy food groups.
Thank you. One small correction: sugar is a food ingredient. It's not a food group. And only if you include now, and that would be a question for clarification, all sorts of sugar sources. But I assume you're talking about the refined sugar grain, yeah? Oh, it's a condiment type of thing, although we are taking a lot of this and we are using it a lot. Stima, there's one more question.
All right, thank you so much for the presentation. What could be the contributing factors to the high prices of food in the urban areas and not in the rural areas? What could be the contributing factors for Tanzania?
Okay, thank you. The likely contributing factor for the high prices of food in urban areas, first is because urban settings in Tanzania, specifically in the study area, is the consumption hub, not the production hub. So most of foods consumed in that area come from from other regions of the country. And by the way, that's the biggest city, the trade city in the country. So there is no production, food production in the region. So the transport logistics, the seasonality and everything are the reason for high prices in urban areas compared to the rural areas, as I said earlier.
Thank you. While we are preparing for the next presenter to come in, one more question, going back to my question. Why do you group vegetables into one group? All vegetables. We are talking here about green leafy ones, the provitamin A-rich ones, the other fruits and vegetables, just because they're sold on the same market, because all the other things are also sold on the same market. What is their reason for grouping them as such? And then you're having the seasonality, no? Because you're arguing about the own production.
Okay, thank you
Use
for
the microphone while the other one is already getting prepared, the other speaker.
Okay, thank you for the good question. Actually, when we collected data, we disaggregated the types of fruits and vegetables. We had 3 types. We had vitamin A rich fruits and vegetables. Also, we had leafy vegetables, and then we collected data on any other fruits or vegetable. But for easy comparison and interpretation, we aggregated all of the fruits and vegetables together.
Challenging the next time you don't group them together because they all have different nutrient properties. I mean, each of these groups have their own idea, and that's why if you always go for the easy solution, we will not move forward in understanding the systems. Let us move forward. So let's hear about the Thank you for applauding and also for the next speaker who is now coming in. It's Wilfried. I tried to pronounce his first name, but he challenged me. Okay, we are talking here now about a soft cheese processing and conservation issue. And thank you very much and welcome to you.
Thank you for the opportunity. I'm Wilfried also from University of Parakou in Benin. And I will be presenting this comprehensive overview of West African soft cheese processing and conservation on behalf of the co-authors that are Matthew, Prof. Linda, Prof. Carol, and Prof. Daniel. Sorry. Now, after talking about vegetables and rice, let's talk about cheese. Here you have the fresh milk, that is the raw material or raw ingredient for making the cheese. And here we have the nice West African soft cheese, bent, strained, or molded. Milk and dairy products are vital sources of nutrition and livelihood for millions of people around the world. But because of its highly nutrient dense, milk is very perishable. And one of the best processing methods to conserve its important nutrients is to process it into cheese. And the cheese making or the processing varies between countries and regions, depending on local know-how and culinary traditions. And in Africa, West Africa, more precisely, the technological know-how is still traditional. The West African soft cheese you can have here is nutritious, good source of protein, up to 36%, fat, up to 27%, minerals and vitamins. It's considered as soft cheese because of its content of moisture, up to 60%. But years and years, the West African soft cheese has attracted researchers and research. But with this comprehensive review, we aimed to summarize current knowledge, the perspectives, and what research is needed to enhance the quality and safety of this West African soft cheese. So, to attain our objective, we conducted a systematic search through different databases such as Scopus, Web of Science, PubMed, Google Scholar, and ResearchGate. We included the papers based on inclusion and exclusion criteria, let's say, to include a paper, it should be a survey or experiment with a specific focus on processing and conservation of West African soft cheese. And there were no restrictions on year of the publication, only if the full test is available, and all the stages of screening has been done by 2 reviewers. The keyword for the search query the keywords were the Wawa, the local names of the West African soft cheese, such as Wawa, Wangashi, Wangashi Gasire, Wangashi, etc. So out of 500 papers we got from the different databases, we included 57 papers that met our inclusion criteria. And what we have found is that the West African soft cheese processing steps are the same throughout, let's say, across the West African countries. So the first step is the warming of filtered milk. The second step is the addition of the coagulant, and mainly here the coagulant is the calotropis leaves that are crushed or pounded. And after the addition of the coagulant, we have the coagulation of the milk. Then the cheese is strained and molded, and we have the final product, our West African soft cheese. As I said, the main coagulant here is the Calotropis procera, and the main ingredient is the cow milk. But we have found in the literature that some researcher has been focusing on alternative coagulants to the Calotropis and partial replacement of the cow milk with vegetable drink or milk. Allow me to use drink here for vegetables drink. And the others here is about the survey and research that focus on purchasing samples. Talking about the milk diversity in the West African soft cheese research, we have seen that researchers have been trying soy milk, coconut milk, tiger nut milk, cocoa milk, and bambara groundnut milk. That are the references to the papers. And what is the effect of the partial replacement of cow milk on the quality of West African soft cheese? Quality here, we looked at the yield, the protein, and the fat. And we noticed that 80% of the papers we included, let's say 7 papers, have recorded impact of the partial replacement of the cow milk on the yield, on the protein content, and the fat content. And the partial replacement of cow milk increased the protein and the fat, whereas the yield decreased. Talking about the coagulant diversity, the alternative to calotropis, we have the citrus lemon that have been tested, Carica papaya leaves that have been tested, Moringa oleifera, Chloridium calcium, stick water, that is here the cereal soaking water. And here are some pictures for those who can't find their way. Same as the partial replacement, the change of the coagulant affects the yield, the protein content, and the fat content in West African soft cheese. As I said at the beginning, the processing is still traditional, and the conservation methods are still traditional. So here we have some methods that are boiling with salt solutions, coloring with natural dyes, sorghum stalks. But it has been found that actually this is only a belief, but it's not scientifically proved. Some immerse the cheese in the whey or the water. Some are drying it, smoking, frying. And interestingly, we found that lemon juice as a coagulant reduces population of Enterobacteriaceae yeast and malts, total aerobic bacteria, and so on. In conclusion, you have found that there are similar processing steps across West Africa, but with inconsistency in concentration of coagulants used that lead to the difficulty of the quality assessment or the prediction of the quality of the West African soft cheese. And there is a growing interest in alternative milk sources and coagulants, but there is a limited report on comparison between the alternative substitutions and full milk in West African soft cheese. And we are recommending to do more research, to conduct more research towards milk substitution and coagulant diversification to ensure the sustainable West African soft cheese production. Thank you for your kind attention.
You Wilfried. So I'm wondering where is our tasting now? Especially the sorghum diet variety, it would be very interesting to try. And lemon versus the other one. Okay, any questions? No, none of you is a cheese eater. You're not eating cheese. It's another product for the East Africans.
Well, I would say cheese from West Africa. Big question concerning this diversity in coagulants. Do you see that rather as an impediment to widespread marketing also in urban areas? Or do you see it as a chance for diversity? Because some of those coagulants are better and are worse, are likely affecting shelf life. So would you go for a standardization, say a recommendation, saying, okay, use this if you can, second priority this, or it doesn't matter?
Thank you for the question. Actually, based on these results, we are conducting research trying to find out the effects of those coagulants on the quality of the West African soft cheese using the consumer preferences and acceptance. So maybe at the end of the research, we can make some recommendation.
Thank you. I'm having a question to the audience. If you go back one more slide, please. No, one more. So there is a lemon juice reduces populations. And he was saying it's a surprise. Any explanations, any assistance for him why this may happen? Oh, let's see what the others are saying. No explanations? What happens here?
Well, if I would be you, then I would quickly check on the pH. Because likely with a strong drop in pH, you have antibacterial behavior.
Okay, let's see.
Yeah, I follow in the same pursuit of Probuke. Lemon juice is acidic, and acidic environments prevent the growth of host bacteria. And that is why we are having less growth of populations of enterobacteria as well as yeast and molds.
Okay, any other explanation? Lemon juice is a conservative, what we call a preservative as well. No, but I mean, there's also the possibility that it actually kills the good ones. It doesn't say anything on this slide. No? What's happening to the good yeast and molds to make a cheese tasty? Because cheese is basically molded milk. Anyone, any idea, any experience yourself if you put lemon juice to sour milk? No? Okay. Yeah, I mean, he has asked for more research on that one, no? So it might be quite interesting to test what is happening with the good ones. If you add lemon juice, it would be quite interesting. For sure, it has a taste implication. By this, I would like to thank all of the presenters. If there's no additional question, also from the online participants. Thank you very much for your patience. I think we are a little ahead, but it allows you to actually interact now with the presenters, online as well as in person, over lunch. You may ask regarding the taste, and we had some fresh cheese yesterday. I don't know whether you like the Austrian way of preparing it. And I see a debate there already, so just attend them. And I would like to invite you also for the upcoming poster session, which is still there, at minimum one tomorrow, and I think one in the evening, where food science and nutritional aspects will also be continued to be discussed. And looking forward to interacting with you at any time, and see you later at lunch, tea, coffee, whatever. Maybe you can send this.
I can't hear you. Any discussions?